Welcome back. We are going to finish off chapter 14, which is glycolysis and the fates of glucose. So this lecture will focus on a lot of sections. It seems like a lot, but it's going to be just a brief overview of these many things that happens after glycolysis. So first we'll talk about section 14.2, the feeder pathways for glycolysis, meaning we have many different carbohydrates, and all of those carbohydrates, or most of those carbohydrates, are actually converted into glucose so they can go through glycolysis and we can get energy out of those. Section 14.3 are the fates of pyruvate. So pyruvate is the last product in the glycolysis pathway. And under anaerobic conditions, we have certain fates of pyruvate that we're going to discuss. Then we'll discuss gluconeogenesis, which is how do we actually make glucose when we're in need of glucose from other molecules. And last, we'll talk about the pentose phosphate pathway. Let's start with a review of glycolysis. Remember that glycolysis comes in two phases: the preparatory phase, which we use ATP, and then the payoff phase, which we gain back 4 molecules of ATP and 2 molecules of NADH. I like to memorize by memorizing the enzymes involved in glycolysis: hexokinase, phosphohexosisomerase, phosphofructokinase 1, aldolase, and triose phosphate isomerase. All right, so hexokinase. It's a kinase, and kinase generally take a phosphate off of ATP and add it onto a substrate molecule. So hexokinase takes in a phosphate from ATP, adds it on to our first reactant glucose, and then we get glucose 6-phosphate and ADP. Phosphohexosisomerase is an isomerase, so we're going to isomerize G6P and create F6P. What about phosphofructokinase 1? Well, it's a kinase again, so we're going to take F6P and then add on to it another phosphate from ATP and create F16BP, fructose 1,6-bisphosphate. Our next enzyme is aldolase. Now, aldolase is responsible for cutting our F16BP in half and creating 2 3-carbon molecules. I like to call them GAP, glyceraldehyde 3-phosphate, and DHAP, dihydroxyacetone phosphate. Our next enzyme is triose phosphate isomerase. So in glycolysis, at this point, we've made 2 molecules of 3-carbon compounds, GAP and DHAP, and then we're going to isomerize one of those. So we have 2 molecules of GAP. So triose phosphate isomerase creates a second molecule of GAP. Now we're at the payoff phase. The enzymes involved in the payoff phase are glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase. So again, we're only memorizing 10 enzymes and then figuring out the reactions from those. So now we have 2 molecules of GAP. We're going to utilize glyceraldehyde 3-phosphate dehydrogenase. A dehydrogenase is an oxidoreductase. Here in our payoff phase, we're creating molecules that have energy potential, such as NADH or ATP. So our glyceraldehyde 3-phosphate dehydrogenase, basically GAP dehydrogenase, is going to take a hydrogen from our GAP molecule and add it on to NAD+ and create NADH. Since we have 2 molecules of GAP, we create 2 molecules of NADH. This enzyme also adds a phosphate group to our GAP molecule, or our glyceraldehyde 3-phosphate. So we're taking an inorganic phosphate. So when we add on an inorganic phosphate to GAP, we have 1,3-BPG, 1,3-bisphosphate glycerate. We have 2 molecules of 1,3-bisphosphoglycerate. At this stage, we're going to utilize phosphoglycerate kinase. Again, another kinase. What do kinases do? They usually take a phosphate from ATP and put it onto the substrate molecule, but in this case, it's named for the first reaction. Remembering we're in a payoff phase, we don't want to make molecules of ADP, we want to make molecules of ATP. So we're going to take off 1 phosphate from 1,3-BPG and create 3-phosphoglycerate. So we remove the phosphate from 1,3-BPG. Remember, there's a reaction in there that's important. We're going to create an intermediate 2,3-BPG. And we create 2 molecules of 3-phosphoglycerate and 2 molecules of ATP. The next enzyme that's utilized is phosphoglycerate mutase. We're just mutating the 3-phosphoglycerate to 2-phosphoglycerate. So now we have 2 molecules of 2-phosphoglycerate. We, in the next reaction, utilize enolase to get rid of a water molecule and create a phosphoenolpyruvate, usually called PEP. So now we have 2 molecules of PEP. PEP is a very energetic molecule that can easily give away that last phosphate and add it onto ADP to create ATP utilizing pyruvate kinase. And in the end, we have 2 molecules of pyruvate. Let's move on to feeder pathways for glycolysis. We learned the structures and the function of many other monosaccharides and disaccharides and polysaccharides, and most of those are going to actually go through glycolysis, but they must be converted into glucose first. So many of our carbohydrates are metabolized actually through glycolysis, but we need to learn how they actually are turned into glucose first. So this figure on the right, it seems like a whole lot is going on. But it's actually really easy if you break it down and see what's going on as we take things like galactose or mannose and convert them either into glucose or some other step within the glycolysis pathway. The ones that I want you to learn are how lactose and galactose actually enter into glycolysis, how sucrose and fructose enter into glycolysis, how glycogen and starch enter the glycolysis pathway, and how mannose enters the glycolysis pathway. Okay, so first let's start with lactose. So lactose is a disaccharide. It's made of glucose stuck to galactose. So first we have to break down that lactose by the enzyme. You already know this, lactase. Let me highlight that. So you drink some milk, you eat some yogurt, your body has to break down that sugar into a form that you can use. So in your mouth and your digestive system, you start breaking down the lactose into its 2 monosaccharides, glucose and galactose. So that's simple. Glucose just goes right into the glycolysis pathway. But then there's galactose. Usually you're going to only eat galactose through the breakdown of lactose by lactase. So how does galactose get broken down? There's a bunch of steps. You don't really need to know all those steps. You just need to know that galactose gets converted to glucose 1-phosphate and then converted into glucose 6-phosphate. The name of the enzyme doesn't really matter, but it's a mutase. And so it's going to move the phosphate on glucose 1-phosphate to create glucose 6-phosphate, and we know that glucose 6-phosphate is the second step in glycolysis. So lactose gets broken down by lactase. So we're going to break apart this lactose by lactase. This is awful. All right, so we broke that apart and we created glucose and galactose. And then from galactose, we create G1P and then G6P. And G6P is in the glycolysis pathway. Okay, so then what about sucrose? You memorize the structure of sucrose, so you know that it is a disaccharide of glucose and fructose. So we get that in our mouths or in our stomach, and then it gets broken down. So we need to break down sucrose. How are we going to break down sucrose? With an enzyme called sucrase. That's pretty easy to remember. Lactase breaks down lactose. Sucrase breaks down sucrose. So we're going to break down sucrose with sucrase. Okay. I don't know if those actually look like words or not. But anyway, so you're going to break that down with sucrase, and glucose goes right into the glycolysis pathway. But what about fructose? Well, actually, fructose has two pathways it can go on, depending on what organ it's being broken down in. But we'll just learn this one, that fructose is acted upon by hexokinase. So you already seen hexokinase. Hexokinase is a kinase, so it's actually going to take a phosphate from ATP and add it onto the substrate. So we're going to utilize hexokinase again. It's good that that enzyme has multiple uses and create F6P. F6P. All right, so we've broken down sucrose into glucose and fructose. Glucose goes through the glycolysis pathway. Now we need to get fructose in. Well, fructose gets phosphorylated to F6P, which is the third step in glycolysis. All right, so then what about glycogen and starch? Glycogen and starch are just polysaccharides of glucose. That's it. And so we can take our glycogen or our starch and break those down by, not that one, amylase. So we can break those down with amylase. Use this one. Because they are just polysaccharides of glucose. We know starch is made of amylose or amylopectin. So in our mouths we have amylase, which breaks down amylose or glycogen too. And in our stomach we have another amylase. So amylose is just broken down by amylase and produces glucose, and glucose, of course, is glucose, can go into the glycolytic pathway. Our last one here is mannose. So mannose gets turned into mannose 6-phosphate by hexokinase, and then it gets isomerized to F6P. So mannose has two steps. So we're going to create M6P. Okay, so we're going to phosphorylate that with a kinase, hexokinase, and then isomerize it to F6P. And F6P is in the glycolytic pathway. So these are the 1, 2, 3, 4 that you should be familiar with. How do we get these things into the glycolysis pathway? We need to either turn them into glucose, or we need to maybe phosphorylate them and turn them into G6P or F6P, which appear in the glycolysis pathway. So what happens to pyruvate after glycolysis? We got actually very little energy from the breakdown of the 6-carbon sugar. So when we create those two molecules of pyruvate, they represent a fork in the metabolic pathway. So there are 3 paths that they can actually take. We'll talk about the entrance of the 2 pyruvate molecules into the citric acid cycle in the next chapter. So if you're an animal, or you are a plant, or some microbes under aerobic conditions, which is with a lot of oxygen, you're going to just enter the TCA cycle or the Krebs cycle or the citric acid cycle, whatever you want to call it. We'll talk about that later. But if you are a yeast, you mix that with some sugar, you get alcohol. So that's called fermentation. We'll talk about how we go from the 2 molecules of pyruvate into making ethanol, fermentation. And we'll talk about under anaerobic conditions, or where there's very little oxygen around, like you're running a marathon or you're working muscles a lot, you're going to convert those 2 molecules of pyruvate into lactate. So usually we want to be under aerobic conditions with a lot of oxygen so that we can get as much energy out of that glucose as we can going into the TCA cycle. But yeast actually ferment that pyruvate into ethanol, or if we are under hypoxic or anaerobic conditions, we convert those 2 molecules of pyruvate into 2 molecules of lactate. Under anaerobic conditions, that means where there's no oxygen or very, very little oxygen, our pyruvate is converted into lactate utilizing the enzyme lactate dehydrogenase. So at the end of glycolysis, we have 2 molecules of pyruvate. In order to go through the citric acid cycle, we actually need oxygen. And during strenuous exercise, the muscles are working so fast and using up so much energy that oxygen cannot be supplied fast enough. So your muscle cells will convert pyruvate into lactate. It utilizes a dehydrogenase, and we remember that a dehydrogenase is an oxidoreductase. So we would generate NAD plus from NADH. And an NAD plus is necessary because it can be used in glycolysis. Now, this process of converting pyruvate to lactate can't go on for very long because the acidification of muscle can be problematic. And actually, you can only do a sprint for about a minute before you actually need to generate energy in a different way. The lactate doesn't give us energy itself, but the lactate is an alternative fuel because it can be transported to the liver, and then it can undergo gluconeogenesis, where we can convert it back to glucose. So if you convert it back to glucose, you can get energy, 2 molecules of ATP, 2 molecules of NADH from that. It's a very inefficient process, so it can't be used for very long, but lactate can become an alternative fuel source. The organism yeast and some other microorganisms actually go through what we think of as fermentation. So when we have wine or vodka or some other alcoholic beverage, it is actually turning that pyruvate into ethanol by their other pathway of getting energy. So they don't go through the citric acid cycle, they actually just go through this fermentation. So after glycolysis, again, we have 2 molecules of pyruvate, and then we utilize pyruvate decarboxylase, and it does what we suggest it does. It decarboxylates. So we take away a CO2. So now we form one byproduct, a molecule of CO2. That creates acetaldehyde, and acetaldehyde then is converted into ethanol. So we go from pyruvate to acetaldehyde to ethanol, utilizing an alcohol dehydrogenase. So this actual reaction is named, or the enzyme is named for the reverse reaction, alcohol dehydrogenase. And there again, we are converting NADH to NAD+. And that way we have regenerated NAD+, because we need that for glycolysis. Yeast have this alcohol dehydrogenase, but we also have an alcohol dehydrogenase, so we can get the reverse reaction so that we can metabolize alcohol. So note the formation of CO2. There's no net oxidation happening, and that's characteristic of fermentation. That hydrogen to carbon ratio stays the same. And the other thing I want you to notice is that both steps require cofactors. So we haven't really seen cofactors yet, but they will be very important in the citric acid cycle. So we utilize a magnesium and thiamine pyrophosphate. This is an important enzyme, and when this enzyme is in short supply, this cofactor is in short supply, you end up getting diseases. So thiamine pyrophosphate, or TPP, those are the same thing. It's a derivative of vitamin B. So if you don't have enough vitamin B in your diet, you cannot have this reaction, and you'll have disease from that. Now we have gone over many objectives. We've gone over glycolysis and feeder pathways of other carbohydrates so they can go through glycolysis. Then we talked about the fate of pyruvate, either into lactate or going to the citric acid cycle, which we'll talk about in the next chapter, or converted into ethanol by yeast. Now we're on our third objective, which is gluconeogenesis, which is the creation of glucose from other precursors. So carbohydrates are essential nutrients. They are molecular precursors to glycoproteins. They appear in our glycolipids. Our brain usually only wants to use glucose as its source of energy. It uses about 120 grams of glucose each day. And so if we look at this chart, we see that lactate can be converted into pyruvate. Pyruvate then can go through gluconeogenesis to create our PEP molecule, our G6P, and then be converted into blood glucose. We cannot utilize the entire fatty acid, but we can take our storage molecule, our triglycerols, and take the glycerol off and convert that essentially back into gluconeogenesis so we can get glucose. And plants can take 3-phosphoglycerate, put that back into gluconeogenesis so they can create glucose. If we don't need glucose, we can convert those to many other things, such as glycogen for storage. Plants can convert it into starch for storage, or some plants can make other monosaccharides or disaccharides, such as sucrose. Glycolysis and gluconeogenesis are very similar in that glycolysis creates 2 molecules of pyruvate, and gluconeogenesis usually feeds in a molecule of pyruvate, and then it goes backwards through glycolysis to create glucose. Glycolysis, though, occurs mainly in the muscle and the brain. However, gluconeogenesis occurs mainly in the liver. These pathways are quite similar, but they're not identical. We don't have just the opposite of glycolysis being gluconeogenesis. There are 3 steps of the 10 steps in glycolysis that are catalyzed by different enzymes in gluconeogenesis. So here, we'll go through all these steps in a minute, but here we have glycolysis. There's 10 steps. Those enzymes that we are familiar with: hexokinase, PFK1, enolase, aldolase, all of those. But some of these are not identical enzymes in gluconeogenesis, and the three that are not are hexokinase, which is an irreversible step in glycolysis, the use of phosphofructose kinase 1 or PFK1. It's an irreversible step, so that enzyme is not utilized in gluconeogenesis. And the last step is different. We actually are going to add a step in gluconeogenesis and not utilize pyruvate kinase. So we're just learning four more enzymes in order to perform gluconeogenesis. We will go over and memorize the three irreversible steps in glycolysis that utilize different enzymes in gluconeogenesis. The first step that is irreversible is the conversion of PEP, or phosphoenolpyruvate, to pyruvate. It's the last step in glycolysis. It utilizes different enzymes in a different part of the cell that give a different pathway for gluconeogenesis. It's a pretty roundabout and complex pathway to get from pyruvate to PEP, so we'll spend a little time on that. The next irreversible step in glycolysis is the conversion of fructose 1,6-bisphosphate, or F16BP, to F6P. That's the third step in glycolysis. We'll utilize a different enzyme for that. So the PFK1 in glycolysis converts F6P to F16BP, and we'll utilize fructose 1,6-bisphosphatase in gluconeogenesis. So to go from fructose 1,6-bisphosphate to fructose 6-phosphate, we utilize 1,6-bisphosphatase 1. And then the first step in gluconeogenesis or glycolysis, which is the last step in gluconeogenesis, to convert G6P to glucose, does not use hexokinase. We saw hexokinase being used many other times in the feeder pathways into glycolysis. But we utilize glucose 6-phosphatase to convert G6P to glucose. The last step in glycolysis, step 10, is to convert PEP into pyruvate. And so the first couple of steps in gluconeogenesis is to convert pyruvate into the PEP molecule. The conversion of pyruvate into the energy-rich phosphoenolpyruvate, or PEP, requires two energy-consuming steps. In the first step, pyruvate is transported into the mitochondria and then converted to oxaloacetate. So we have our pyruvate molecule here. We utilize pyruvate carboxylase, so we're going to carboxylate pyruvase using bicarbonate. So we're going to take that CO2 over bicarbonate and create this molecule, oxaloacetate. So it's good to know that oxaloacetate is going to appear in the citric acid cycle. So we can go ahead and memorize oxaloacetate. So the first half step really in gluconeogenesis is to add a carbon dioxide to pyruvate and create oxaloacetate. Now that we've created the oxaloacetate, we can convert that to our phosphoenolpyruvate, our PEP molecule. So to add a phosphate to that, we're going to utilize Pepcarboxylase. So kinases usually take a phosphate off of an ATP, but now we're taking a phosphate off of GTP and adding it to oxaloacetate and creating our PEP molecule. So our first step in gluconeogenesis to go from pyruvate to PEP actually occurs in two phases. First, we create oxaloacetate, then we phosphorylate the oxaloacetate by PEP carboxykinase to create phosphoenolpyruvate. The PEP carboxykinase reaction occurs in either the cytosol, the mitochondria, but uses different enzymes. This is a reversible reaction under cellular conditions, And the formation of one high energy phosphate compound, the PEP, is balanced by the hydrolysis of a GTP. To summarize, in order to convert pyruvate to PEP, we need 2 steps. And the net equation is that we have pyruvate, we must invest 1 ATP and 1 GTP and a bicarbonate, and then we get PEP, ADP, GDP, an inorganic phosphate, and CO2. The other 2 steps in gluconeogenesis that utilize different enzymes than glycolysis are the conversion to F6P and G6P. So once we are going along with gluconeogenesis and we have created fructose 1,6-bisphosphate, instead of utilizing PFK1, we utilize fructose 1,6-bisphosphatase, and it hydrolyzes the phosphate at C1, releasing an inorganic phosphate and creating F6P, fructose 6-phosphate. And then the last step in gluconeogenesis to create glucose goes from glucose 6-phosphate, utilizing glucose 6-phosphatase, and hydrolyzes the phosphate at the C6 position to produce glucose and inorganic phosphate. Here we have a side-by-side of glycolysis on the left and gluconeogenesis on the right. So glycolysis or gluconeogenesis is the reverse of glycolysis, utilizing the exact same enzymes for most of the reactions. However, the irreversible reactions utilize a different enzyme. So we can notice the net equation for glycolysis. We input 1 molecule of glucose. We have to input 2 molecules of NAD+, 2 ADPs, plus 2 inorganic phosphates, and we get out 2 pyruvates. We get the molecules with a lot of energetic potential, 2 molecules of NADH, 2 ATPs, and 2 water molecules. And it has a net delta G of negative 60 kilojoules per mole. But if we look at gluconeogenesis, we put in 2 pyruvates. We have to use 4 ATPs, 2 GTPs, 2 NADHs, and 4 water molecules. But we get back 1 glucose, 4 ADPs, 2 GDPs, 6 inorganic phosphates, and 2 NAD pluses. So it's energetically very expensive, but it is very necessary because some certain organs, especially our brain, actually only try to utilize glucose for their energetic needs, unless of course you're on some stupid keto diet. Our last objective is to understand the pentose phosphate pathway. So there are a lot of steps here in the pentose phosphate pathway. You don't have to know any of them. I will tell you what you should know conceptually about the pentose phosphate pathway. So if you remember, the first step in glycolysis is to create G6P from glucose and ATP. This is not a reversible step. So once G6P is created, it has to go through the glycolysis pathway. But alternatively, glucose 6-phosphate is used in the pentose phosphate pathway. And what is the pentose phosphate pathway? Well, we're making a pentose. So if you remember the structure of our nucleotides, one pentose is very important, and that is ribose. So from G6P, we can make ribose 5-phosphate. You see here that we've lost a CO2. Of course, you have to lose a carbon to go from a 6-carbon G6P to a 5-carbon ribose 5-phosphate. But in a lot of steps are involved in it. You don't have to know all of those steps. But it is important for you to know that once we've created G6P, it has an alternative pathway than the glycolysis pathway called the pentose phosphate pathway, where we lose the CO2. We've gone from a 5-carbon sugar that's phosphorylated to a 6-carbon sugar. A 6-carbon sugar that's phosphorylated to a 5-carbon sugar that's phosphorylated. And this ribose 5-phosphate is important because it is incorporated into our nucleotides that is necessary to make our DNA and our RNA. Other important notes about the pentose phosphate pathway are that the main products are NADPH, so we created a molecule with energetic potential, and ribose 5-phosphate. NADPH is an electron donor, reductive biosynthesis of fatty acids and steroids. It's used for repair of oxidative damage. And ribose 5-phosphate is a biosynthetic precursor of nucleotides. So we need that to attach to our nitrogenous bases so that we can make RNA and DNA or the synthesis of some of our coenzymes. Very, very briefly, I want to go over glycolysis and gluconeogenesis regulation. This is kind of an overall summary of chapter 15 in the next couple of slides. So we must keep a certain homeostasis. We must not make too many products or too many metabolites, and we need to regulate when these things need to be stored or when they need to be used or when we need to make more of maybe our energy molecules. So there are certain steps in glycolysis and gluconeogenesis that need to be regulated. And the regulated steps are the same ones that we see are different in glycolysis and gluconeogenesis. So that would be the step 1, the conversion of glucose to glucose 6-phosphate. That would be the conversion of fructose 6-phosphate to fructose 1,6-BP, and also the conversion of PEP to pyruvate. The first step in glycolysis is catalyzed by the enzyme hexokinase. It's regulated by the fact that there are 4 isozymes of hexokinase. Isozymes are different enzymes that catalyze the same reaction. They typically share similar amino acid sequences, but their kinetics and resulting regulation can be different. Hexokinase regulation involves tissue-specific forms of the enzyme. So hexokinase 1 through 3 is usually found in the muscle and the blood cells. It has an extremely low Km. That means that in specifically muscle cells that have high energy needs, hexokinase is normally active and operating at its Vmax because the Km is much, much lower than the concentration of glucose. But it is allosterically inhibited by G6P, its product. So once G6P starts to rise up too much, it reversibly inhibits hexokinase 1 through 3. Hexokinase 4, however, also called glucokinase, is found in the liver. The Km is about 10 millimolar. So it doesn't operate at its Vmax until the blood glucose levels get to be extremely high. So it remains active at extremely high concentrations of glucose, and if the glucose gets to be too high, that glucose can be transported to other cells. The second step in glycolysis utilizes the enzyme PFK1, and the corresponding step in gluconeogenesis utilizes FBPase1. Once F16BP is created, it's a committed step in glycolysis, meaning if you create G6P, G6P can go on to other pathways to creating amino acids or creating ribose, as we've seen in the pentose phosphate pathway. However, F16BP is a committed step in glycolysis. Once you make that, it has to go through the glycolytic pathway. So this step is very regulated on many levels. I'll just talk about one level that it is regulated. So if you need energy, of course you want to go through glycolysis so you can create ATP or NADH. But if you have enough energy, you want to go through gluconeogenesis. That way you can create more glucose, and that glucose can be stored as glycogen so it can be used later. So when you need energy, you have high concentrations of AMP, high concentrations of ADP, but low concentrations of energy, ATP. If you have plenty of energy, you'll have low concentrations of AMP, high concentrations of ATP, as well as high concentrations of citrate, which will appear in other pathways, such as the citric acid cycle. So if we look at this figure here, we see that we created F6P, and PFK can convert that F6P to F16BP. But ATP is an allosteric inhibitor of PFK1. Citrate is also an allosteric inhibitor of PFK1. But when we have low concentrations of high concentrations of ADP and AMP, that replaces ATP in inhibiting PFK1, and we have an active PFK1. And at the same time, AMP inhibits FBPase1. If you have a lot of AMP around, it means you need energy, so you don't want to go into making glucose. You want to go in through the glycolysis pathway and get rid of that glucose and turn it into energy. Our last regulated step in glycolysis regulates pyruvate kinase. So we see here that in signs of glucose depletion, means there are low concentration of glucose, our hormone glucagon gets released. So we release glucagon, and through a cascade of events similar to ones that we saw in chapter 12, PKA, protein kinase A, gets activated, and then it phosphorylates pyruvate kinase L. So there are two isoforms of pyruvate kinase. One is just in the liver. So when glucose is low, we inactivate pyruvate kinase L, and that allows more glucose to build up. So the liver, instead of breaking down glucose for energy, it can actually break down glycogen to release more glucose into the bloodstream. In all other tissues, including the liver, we see allosteric regulation or inhibition, allosteric activation or inhibition. So we see when there is alanine, a high concentration of alanine, we inhibit pyruvate kinase. Well, alanine is just a product of the transamination of pyruvate. So high concentration of alanine means high concentration of pyruvate, and that inhibits pyruvate kinase. If we have high concentrations of acetyl-CoA, which we'll talk about in the citric acid cycle, or high concentrations of fatty acids, which means we have a lot of energy, they also inhibit pyruvate kinase. But if we have abundant F16BP or abundant ATP, we actually help to activate pyruvate kinase. So now to summarize the regulation of glycolysis and gluconeogenesis. The main purpose, of course, of glycolysis is to produce energy. We primarily want energy in our muscles, our kidneys, our liver tissue, or wherever energy is needed. We activate glycolysis when energy supplies are low. So if there are low concentrations of ATP, we need more. High concentrations of AMP, that means we've used up ATP. Low concentrations of acetyl-CoA or citrate. Glycolysis is inhibited when energy supplies are high or glucose is scarce. When ATP concentrations are high, AMP concentrations are low, acetyl-CoA and citrate concentrations are high, and we'll see that in the citric acid cycle. That means that we are metabolizing. But the main purpose of gluconeogenesis is to maintain glucose homeostasis. We mainly see gluconeogenesis in the liver. Liver is the glucose storehouse. We see glycogen stored in our liver. And gluconeogenesis is inhibited when glucose levels are high or energy is low. So when ATP is low, but also when F26BP is high and insulin is high. We see gluconeogenesis activated when glucose levels are low or energy is high. So we activate it when ATP is high and AMP is low, acetyl-CoA is high, citrate is high, and F26B and glucagon concentrations are low. To summarize what you should have learned from chapter 14, you should have learned all the steps of glycolysis, the 10 enzymes, and many substrates and products. And this is done under anaerobic conditions, meaning there's no oxygen necessary for glycolysis. We just get energy out of glucose, and we turn one molecule of 6-carbon glucose into 2 molecules of pyruvate. And then we also learned that pyruvate has other fates under aerobic conditions. They go on to the citric acid cycle, but under anaerobic conditions, we create lactate or ethanol if you're a yeast or another organism. Then we learned about feeding into glycolysis, how other carbohydrates or disaccharides or polysaccharides are converted into molecules that enter the glycolysis pathway. Then we learned about gluconeogenesis, a process by which cells can use a variety of metabolites to synthesize glucose. And lastly, very briefly, you should understand some of the important aspects of the pentose phosphate pathway, which is the process by which cells can generate reducing power. They generate NADPH that is needed for the biosynthesis of various compounds. And we generate 5-ribose 5-phosphate, so we can incorporate that into our DNA and our RNA.